Today, we are honored to have with us Dr. John Ryals, a visionary scientist and entrepreneur with over 30 years of leadership in biotech. He is the founder and past CEO of Metabolon, the global leader in metabolomics, and the founder of Paradigm Genetics, a pioneer in agriculture biotechnology. Currently, he is a co-founder and board member of AgBiome, a company that uses soil microbes to protect crops while reducing pesticide use. From his early curiosity about the world to switching majors from music to science to launching groundbreaking companies that changed biotech, Dr. Ryals has lived an extraordinary journey of discovery. Today, we'll trace that journey and learn what's next in the future of science. Dr. Ryals, welcome to the BCI podcast.
Thank you.
Let's have a deep dive, So tell us more about your childhood days. Where did you grow up as a kid?
Well, I was born in Waco, Texas, 1954. I was definitely a baby boomer. My dad had been in World War II and came back and pretty quickly met my mom and married her. And, and he was from Waco. So I lived in Waco. It's a nice little town. I don't know if you've ever been there, but it's a very nice town to see. It's small and it's, maybe a couple 100,000 people. Anyway, my dad was an engineer, so he was a civil engineer. And so he moved us to Dallas when I was 6. And then I grew up in Dallas. And so what was I like as a kid?
Yes, like a curious kid, somebody who loves science just tell more about your childhood. Yes.
I was quizzical. I was rambunctious. I wasn't afraid to do things, and I got in trouble a lot. A bad recipe.
Bad recipe. But looking back, do you regret to be bad or it was fun?
It's fun.
This is fun.
Loved my childhood. Yeah.
Fantastic. But Dr. Ryals, when was it that you thought that you'll pursue science?
Well, I didn't know what I was going to pursue through high school. So I played in a garage band. We wanted to be the next Beatles. This was the 60s. And, I just thought I needed a degree. So I went to a community college and got a triple major in music, biology, and science. And then I went to a local university it's now called the University of North Texas. And actually, I was on the board of that. And that was a very big music school. It's well known. It produced Doc Severinsen and many famous musicians at the time. I went through music school and I didn't really like music theory and all that stuff that you had to learn. Mm-hmm. I was just interested in playing. So I dropped the music major and didn't really drop being a musician yet, but a couple of years later, I went to a Bee Gees concert in Fort Worth, and the Bee Gees were really a great group in the 60s, and I loved their music, and I thought, they're going to play that. But they came out and played disco, and I thought...
So you were disappointed.
My God. And then disco started opening up everywhere, and I just thought, this isn't for me. So anyway, I started to focus on science. I wasn't sure exactly what kind of science. I didn't want to go into medicine, and that was the conscious decision, because one time I was about 20 and I went home because I had a bad, and every 15 minutes my mother would come in there and said, do you need anything? Do you need anything? And I finally said, I need you to quit coming in here.
Like any kid, yeah. She was bothering you.
Yeah, and then I thought about it, and I thought, I'm not going to make a very good doctor.
Just because of the flu?
Yeah, I just, so anyway, as I graduated, looked at, I had an offer from Southwestern Medical School to be a physician and a couple other places. And I just thought about it. I thought, no, I don't think I want to do that. And you had asked me about these 2 books in back of me. So the molecular biology of the gene and the Ascent of Man. So during that time, I don't know if you ever saw the Ascent of Man on PBS.
No, I didn't.
It was by a guy named Jacob Bronowski, and he was a very famous guy. And he just went through the archaeology of man, and it was very impressive. And so I bought his book, and then I bought the other book behind me, Molecular Biology of the Gene.
That I know, yes.
Yeah, and that was written by Jim Watson.
Hmm.
And that really encouraged me, you know. I thought, you know, that's what I want to do. And so go ahead.
No, I just was curious. So it never occurred to you during your bachelor's in the college, in the community college, that you would go and pursue science and become a scientist. It was only after you were
Not
like forced to decide.
right. You know, I was thinking of the career a scientist would have, you know, and it's like, okay, I'll have to be a professor, and you know, that's not a bad job. But anyway, I went through the process and things, and a friend of mine, gotten I'd gotten acceptance at Baylor College of Medicine, at University of Texas at Dallas, at University Southwest Medical School, and at LSU. And I was married at the time, and my wife really wasn't about wanting to move, you know. And so a guy told me, he said, "What do you really want to do?" And I said, "I want to do molecular biology." And he kind of kicked back and he said, "Well, I know the place you need to go. You need to go over to the University of Texas at Dallas." And so I applied over there, and I went in for that application, and the guy was Dr. Dimitri Long, who I didn't know it at the time, but he had worked with Kleinschmidt, the guy who developed electron microscopy of macromolecules. But sitting on the wall was the picture of the DNA coming out of the phage. It's a very, very, very famous picture.
Hmm.
And it proved that, you know, phage was one single molecule, and you could trace it from head to tail. And I looked at the picture and I thought, and I asked him, I said, where can I get one of these? What store did you get it at? And he said, I took that picture. And then I
Wow.
shut up. Yeah, I kind of shut up. It's like, wow, he took it. And he told me this story of taking it. And I thought, Jesus Christ. And then he took me around and I talked to various other people. Well, it ended up that institute had been a national lab. And the CIA had used it as part of the German brain drain. And so they collected a bunch of German scientists in Oak Ridge, and then they needed a place to put them. And so they put 16 German molecular biologists in this one institute. And it was run by a guy who was one of the heads of the RAF, and there was one American.
Tell me, like, the whole context, like they were like Nazi professors.
No, they were
No?
Nazi youths. You know, most of them had not finished their degrees, and they rejected Nazism. You know,
Okay.
and they got out as quick as they could. But, you know, and as the war was ending, the U.S. was rapidly trying to get the professors out of Germany because it knew that if they didn't, Russia would get them. And so they developed these, you know, national labs all over the place, and that would be the place they put them. And that's the school I was in. So 16 German scientists. And these guys were not nice guys.
You mean tough guys?
Yeah. And anyway, I talked to all of them. One of the guys was the guy that discovered plasmids. And I thought, wow, that's pretty cool. And he had a plasmid on the wall. They all showed off. And by the time I left, I thought, wow, this is a great place. I want to come here. And I went back to Dimitri's office to say bye, and he gave me an offer. Yeah.
An offer as in for master's, or you're directly for a PhD? How was it?
Well, as a graduate student headed towards a PhD, and with full stipend and all that, full ride. And actually, and it's a very, very long story, I won't go into it, but that happened, and it really shocked me because I'd never heard of anybody getting an offer while they toured the labs. And so,
Yeah.
but in my early days, I was tracked by the CIA as an intellectual. I don't know if you knew they did that in the 60s. I
But
didn't know
why?
they.
Because of your
Well,
German connection.
no, I wasn't German.
No, like, because of these professors who were German, like this 16
No,
German
this was
professors. No?
happening when I was in junior high and high school.
Okay.
And it was a long story, but the CIA was conducting an experiment in Dallas, and they were trying to group all the smart people into a small class,
Mm-hmm.
and everybody else was outside. And so I was in what's called honors. And when I was a rising senior, I was called into the principal's office, and there's two guys in suits there. And I'm like, what? And they said, we want you to join the CIA. And I said, no, one thing I'm not going to do is join the CIA. Anyway, it ends up, because one of my friends did take that.
Mhm.
And it ends up, once you have been offered a job, they keep your name forever. And so they had a role in getting me into UTD. I don't know what it was, but I'm pretty sure.
So that's why you think you got the offer straight away.
Yeah.
Okay.
Yeah. And I had good grades and I had high
Mhm.
IQ and everything. So, and I accepted it right away. And it was the only department of molecular biology that really existed at the time. I mean, Berkeley had one, but that was it. So, that's what happened. And I started going to the university.
And any particular reason that you kind of pursued, one like this topic, like of bacteria, like you said about the viruses, right? Because you were fascinated by the virus, right?
Well, by that picture, but they take you in your first year through everyone's lab, and you serve in the lab for about a month. And I did that. And the guy that I was most impressed with was a guy named Hans Bremer.
Hmm.
And Hans was a bacteriologist and fantastic mathematician. And he had developed the mathematics behind how bacteria grow, but he needed to get the evidence. He had
Hmm.
the equations. And so I got into it, and my job was to do this study on a thing called, God, can't even remember what it's called anymore. And I got into it and saw that what was happening was that when bacteria started slowing down, they started making guanosine tetraphosphate.
Hmm.
And, and ribosome makes that. So an
Hmm.
uncharged ribosome, it attaches, uncharged tRNA will attach to it and it's got to get rid of it. And to do that, it uses ATP and converts it to guanosine pentaphosphate, which then degrades into guanosine tetraphosphate. And I showed that that was the molecule that interacted with RNA polymerase to explain to RNA polymerase how the bacteria was growing and what it ought to be doing.
Fantastic.
And I published 10 papers as a grad student. And when it was kind of unheard of. And when I gave my dissertation and everything, then I was supposed to go back and talk to my committee. And I talked to my committee and they said, well, of course you passed. I didn't
With
think they
super
were going to
grades,
fail
yeah.
me. I didn't really think they'd fail me. But then they said, Roy Klaus said, well, do you have any options on where you're going to do a PhD? And I said, a postdoc. And I said, yeah, I've got 3 offers already. One is at University of Cambridge and, with this guy. And the other is at Albert Einstein University with this other guy who is pretty famous. And the other was at Baylor College of Medicine, which at that time was a pretty famous school.
Even now, I think it's quite
Yeah.
famous. Yeah.
And back then, people, especially older men, would slam their hand on the table. And he slammed his hand on the table and said, no, you're not going to do that. You're too good to do that. And I'm like, I thought Cambridge was okay. Why am I too good for Cambridge? And he said, no, no, no. He said, what is the most exciting thing you've seen happen in the past year that you'd really like to go follow
Hmm.
up on? And I said, well, I guess it's the cloning of interferon. And he said, who did that? And I said, it was a guy named Charles Weissman.
Hmm.
And he slammed his hand on the table and said, you mean Charlie?
Yeah.
Swear to God.
Wow.
And I said, yeah, I didn't know they called him Charlie. And he said, you go get your resume. I'm going to send it to Charlie. He had to do it by telephone. What do they call that back then?
Telegram or
Hey, honey, what do they call sending a paper through the telephone back then? Fax. Yeah, it was a fax.
fax? Okay, telefax. Yeah.
And he did. And the next day I get back to school and he calls me in his office and he said, you got a new offer.
From Charlie.
He said, it's from Charles Weissman.
Wow.
And he says, didn't need to see you. I told him what kind of student you were and told him about the papers and everything. And he said, yeah, I'll take him. And so he was at the University of Zurich.
Mm-hmm.
We had just had a baby,
Mm-hmm.
so I couldn't fly right away. And Weissman came to give a talk at one of the first biotechnology conferences in Houston, and
Mm-hmm.
he wanted me to meet him. And so I drove down there and we had lunch. And he asked me some questions. I didn't realize I was taking a quiz. I was answering his questions. And he said, shit, I can't remember. He said, well, I'll think of it in a minute. But he asked me this question that was kind of tricky.
Mm-hmm.
And I knew it was kind of tricky and I knew the answer. And I said, well, you'd clone it this way. You couldn't clone it that way because you'd pick up everything around it. And he said, you're right. How soon can you be in my office?
Lab or office.
Yeah, office lab. And I said, well, we've got this young child. She's 3.5 pounds. Well, she wasn't even that weight. She's got to be 3.5 pounds before we can fly.
Mm-hmm.
And so we just got to get her to grow. And by December of that year, she had she had to be 5 pounds. And so she attained 5 pounds. And January 1st, we bought tickets to Zurich and flew there. It's the first time I'd ever been on an airplane. And same for my wife.
Wow.
Yeah. So anyway, we get to Zurich and, it was Switzerland. It looked just like it says.
Yes.
And so, I got in there and I was number 30 of 30 postdocs. And, so I was at the bottom at the start.
Wow. But he had 30 postdocs during those times. So huge lab.
Huge lab. And plus he had a lot of Biogen. He co-founded Biogen. And then they founded it on the cloning of interferon. And so, yeah, he had a lab of 30 guys. Some of them were from Biogen. And he got us an apartment paid for by the university, near the university, and we got into it and got the baby settled. And then I went in, but I had to be there with my wife to make sure
Everything was okay, yeah.
Neither of us spoke German, and Swiss Deutsch isn't really German. Anyway, I get to the lab and everybody's speaking English. And I told him I was there, went into his office, and he said, the project I was talking to you about, because he wanted to clone the splicing gene.
Mm-hmm.
And that was the question. He said, if you were going to clone the splicing gene, how would you do it? And, this was in the very early days of cloning. And I said, well, you'd have to isolate the messenger RNA. Then you'd have to put in the splicing gene, but you would look for a product that got bigger and not smaller.
Mm-hmm.
And you'd have to understand all that. And I did. It was his trick question.
Wow.
And I said, once you had an assay, then you just use the assay to purify the enzyme. And there you go. Make antibodies, go get it off a plate. And he said, that's right. And the way he acted, I didn't really, I realized at the table that was a question. So anyway, I got there and he said, I thought about your project. This is what I want you to do. I want you to take over this work from this guy. And what he's doing is he is mapping the beta globin gene promoter.
Mm-hmm.
It was the mouse beta globin gene promoter. And you did that by promoter bashing at the time. I mean, promoter bashing had just been started. And so, I go to the lab, we do that, and we isolated a fragment that ended up being about an 8-molecule fragment of the promoter that
Mm-hmm.
worked like an inducible, I can't think of that. It'll come back to me, but.
So it's like the fragment of the promoter region, right?
Yeah, and it's like you could flip it around, you could do stuff like that. So
Mm-hmm.
there's a name for that.
You mean origami? Origami or some?
No, but anyway.
Anyways, yeah, we can.
That's what it was. It was an enhancer. It was a inducible enhancer. And nobody had ever seen that, we thought, until we went to publish. And we published a paper in Cell along with Monty Otis's lab. And he had done the same thing.
Wow.
So we co-published. And that was a little bit of a thing. But then I was in good with the boss, because I had the molecular.
John, this was a big finding, right? Like you were among
Yeah.
so you were among the pioneers who kind of basically discovered enhancers or
Well,
inducers.
yeah. I'd say Walter Schaffner was the guy, and he was
Mm-hmm.
working down the hall. But yeah, it was an interesting time. And, so we did it, and then we put in other promoters and showed that it would, you could put it in a different promoter, and then you could put in the RNA polymerase and the signal, and it would transcribe
Got it.
that promoter. Yeah. So
Wow.
it was a great story. And, we finished that, and then he had me teaching a lot of the scientists and working on their projects. And he needed me to go give the interferon talk one time,
Mm-hmm.
and it was in Cambridge. And I was there, and I gave his talk, and the guy right after me was a guy named Jeff Shell. And Jeff Shell was a plant scientist.
Mm-hmm.
And he was like the father of plant molecular biology. But I had father of plant molecular biology, but I had no idea about plant molecular biology. And I listened to his talk, and he's talking about genetically engineering plants.
Mm-hmm.
And I thought, that's what I'm going to do.
Wow.
And you, as a career guy, you gotta realize there's going to be luck, and you got to take that luck and run with it. You can't just question it. And there's going to be crazy circumstances, which was one like that. Well, anyway, I got back to the lab, and my lab mate had gotten back from interviewing for postdocs. And I said, well, how did it go? And he told me the places he visited. And I said, I just saw this talk. And, that was a time when alpha interferon had failed its first clinical trials. And so I thought, oh, shit, I did all this work, and that's not going anywhere. It ended up it did.
Mm-hmm.
At that time, I was just thinking, I'm going to have to change fields. And I told my lab mate, when he got back, he told me the places that he interviewed, and I said, well, what I really saw for the first time, at this thing I had to do in Cambridge, was this guy who was cloning genes and putting them into plants, and using an agrobacterium. And I said, it's kind of amazing. And here's where the luck comes in. And he says, well, my aunt is the new head of human resources for an agricultural biotechnology unit they just started in Research Triangle Park, and it's led by Mary Dell Chilton.
Mm-hmm.
And I thought, okay, could you get me could I call her? And he said he'd see, and he called his cousin, and his cousin said she's going to be arriving in Zurich the next morning, and you could meet. She said she'll send her a signal, and you can have a coffee with her at the airport. See, these things don't ever happen.
Yeah,
right? And so I did. I met her, and we had coffee, and she told me what she did, and I told her what I had done, and she said, "Oh, well, that's very interesting." And she said, "Well, why do you want to do plants?"
Exactly.
And I said,
my question was the same, like, of course you were fascinated, but you would change a complete different model system, right?
Yeah. And I said, well, my parents and all my cousins are farmers, and if there's something I can do to help the farmers, I'd do that in a heartbeat. And she said, okay, we're going to have dinner in Basel tonight with the
Coffee
head
to dinner,
of
yes.
with the head of the agricultural division. Can you join us? And I had a car, and it's like an hour drive from Zurich to Basel. And I said, sure, tell me where to be. Tell me when to be. And I was there, and I met this guy who's Heinz Geispieler. And he was head of research for the entire ag division, Z. Begeige. And he talked to me for a few minutes, and he said, what lab did you come from? And I said, Charles Weissman's lab. And he nodded his head, and he said, we want to hire you. And two of Mary Dell's lieutenants were sitting at the table, and they said, oh, no, that's not the process. You can't just hire him. And Geispieler looks at them and says, we're hiring him.
Wow. So you had this many times in life, right? Right from the graduate student to kind of, also here, like Very easy.
It's a big part, right? And you got to realize when luck happens, right? Because
Yes.
some
Chance
people
favors
don't
the prepared
realize it,
mind. No,
right?
absolutely. Absolutely.
So anyway, they gave me a job, and I went down to North Carolina where they had built the first agricultural biotechnology research center.
Mhm.
And there were about 50 people, and I was one of 50. Mary Dell Chilton ran it, and she was one of the founders of Agrobacterium. And when I got there, she said, "Why don't we go to dinner at my house?" And I said, "Sure." And she had leftovers. What the hell? You're not out of Texas yet. And at the table, she said, "Now, really, why did I hire you?" And I said, "I'll tell you 2 good reasons. I've met everybody you got. None of them is a molecular biologist. You got some biologists, you got some microbiologists, and you got some chemists and biochemists, but you don't have a molecular biologist. I'm the first one. And so you're going to need me for that." And I said, "But the real reason you're going to need me is I understand Swiss and how the Swiss tell people something." And that was a lesson I'll never forget.
Please tell us. Please tell us. Yeah.
Well, in Switzerland, they never tell you what they want you to do. But anyway, I joined. She didn't know what I meant, and yeah, and the story's pretty funny. But she didn't know what I meant by that. And she said, "Well, we're going to meet with Dr. Geisbuehler here in a couple of weeks, and I want you to be there." And I said, "Okay." And I watched Geisbuehler, and I can read people. One of my talents, one of the things that has helped me succeed is in summer jobs, I was selling encyclopedias door to door in rural Texas. Well, anyway, I meet Geisbuehler and everything, and he's typical Swiss. Now, of course, he was a general in the Swiss Army, because everybody in Switzerland has to serve. He talked to me and talked to me and talked to me. And anyway, he hired me. And, I went in to talk to Mary Dell. I said, "But Mary Dell, I told you, you need a molecular biologist." And then I said, "And you need Swiss interpretation. You need to understand what they're saying to you. They don't act like Americans." And she didn't know what I meant. Didn't know a thing about what I meant. And so at every dinner, when Geisbuehler was there, at the end of dinner, I'd already seen his method, and he would not tell anybody what to do. He would insinuate that there was this issue that needed to be solved, and he expected them to go solve it. And that's the Swiss. The Swiss
way
don't
of.
tell you what to do.
You have to understand what needs to be done.
Right. And so I went to a bunch of dinners with Geisbuehler. So I worked for Ciba-Geigy for 13 years, and I was stuck on a problem that people laughed at me about, but I didn't pick it out. Ciba-Geigy did. And it was a system called systemic acquired resistance in plants.
Mm-hmm.
And it's like an induct resistance. A pathogen infects the plant, sends out some sort of signal, and then other pathogens can't attack. So the plant's defense mechanisms have been alerted. And nobody knew how that worked. And so they put me on it because I'd worked on interferon, and serious to God, Mary Dell thought interferon might be in plants. And I said, "Mary Dell, that's not how it works. There's no interferon in plants." But anyway, I kept going to these dinners and kept watching Geisbuehler do these things. And after about the third dinner, I said, at the end of the conversation, I said, "Dr. Geisbuehler, I want to ask you a question." And I said, "You're always indirectly telling somebody what to do, but you never tell them what to do." I'd solved this problem that nobody knew anything about. They just thought it was magic. And I showed them it was plant innate immunity, and they had compounds that would induce this. So, now they knew what it did, but it, took 10 years. And over that time, I got really close to Geisbuehler. And anytime he was in the United States, he might fly me out to Chicago or somewhere, and we'd have dinner. And every time we'd have dinner, at the end of the dinner, I would ask the question again. I said, "Why don't you tell people directly what you want them to do instead of be indirect and everything? Is that just being Swiss polite?
Mm-hmm.
Or is that because you want them to develop? And he'd never answer. He'd just change the subject. I asked him that question probably 30 times. And then finally he retired, and I went to his house for dinner. And at the end of dinner, I asked the question again.
And did he answer?
No. I knew what the answer was. It wasn't being polite. I mean, he gave them a problem. He wasn't going to give them the solution, and he expected them to get the solution and solve it, you know? And I use that technique nowadays, you know, because it's like, that's pretty clever.
And you think it's effective?
Well, I wasn't as clandestine as he was, you know, but like I took that style, and person would come to me, show me something and say, tell me what to do. And I say, it's not really my job to tell you what to do. It's your job to solve the problem and do it right. And if you do it wrong, I'll give you 2 chances. And that was that's how I ran the company. And you got 2 chances. 3rd chance, you're out. Because that means you're not very smart. You know what I mean? Now I know exactly what Guy Spooler was doing. He really taught me a great deal. But anyway, the deal Ciba merged with Sando, and they created Novartis. Ciba was a heavy science company. Sando was a nose in the butt company. And I got out of Novartis pretty quick, within a couple of years. By that time, I was head of worldwide agricultural research for Syngenta. And, you know, I had a pretty big position, but it's like, this isn't the thing I bought into, you know, so I'm not going to stay here.
So, but John, like just going back, sorry to interrupt. So you were with Guy Spooler, you were working there in a safe job. So when did the next change happen?
Well, Guy Spooler retired.
Okay.
And Ciba and Sando merged to become Novartis.
Okay.
Which I didn't like. But in Novartis, I was the worldwide head of agricultural biotechnology research. Marydale had stepped down, but I was not liking what I was seeing. And then they merged again with someone who became Syngenta, and I was already planning to leave. You know, and I had just looked around, talked to my guys. I had a 20-person lab myself. We were all friends, and I said, guys, you know, I'm going to probably get out of here, but if I do, you know, all of you can have a job. Don't worry about it. Then I had 3 co-founders who were all good friends of mine and worked in my lab. And told them, and they said, yes, this is what we got to do. So let's get the hell out of Dodge, you know, and go form a company. And so on one day, we just all walked out and went to a diner and sat down and said, well, okay, well, what are we going to do? And the next day or two, I was reading the annual report of Monsanto. And I knew the head of research at Monsanto very well. I'd known him for dozens of years. His name was Rob Fraley. And Rob put out a piece of what the future of agriculture would be. Monsanto had already done all the genomics. I mean, they had sunk $500 million into genomics, so that you're not going to battle them out on that. And he said, but the new frontier will be understanding gene function. And, you know, it's like, I got to a whiteboard with a pen and I drew out the central dogma, right? From genes to messenger RNA to proteins to biological proteins. And I said, you could be at genes, but we only know what about 1,000 genes of 25,000 do. And that's still the case. And that was 30 years ago. And I said, messenger RNA is the same way. Proteins are the same way. And plus proteins have secondary control. So just looking at a protein isn't going to say it's active. And I said, but if you go down the next step to metabolism, we know what metabolism is. You know, it's all completely been mapped out. All we have to do is create a technology, you know, where we can measure all the small molecules all at the same time, the way the DNA sequencer does with DNA. And that's what we need to do. And then we need to identify those molecules. And so that set us in. We started it in paradigm genetics, but it was pretty much impossible because machines couldn't handle the amount of data. But Paradigm Genetics, what it did was also knock in and knock out every gene in the in the small plant called Arabidopsis.
Mm-hmm.
And then
Arabidopsis, yeah.
we created lines, we sequenced everything, and then we phenotyped them, because the phenotype is what you're looking for.
Mm-hmm.
And we could score 145 different developmental phenotypes. And so we mapped every gene to a phenotype, and we had that in a database. Well, your investors, and this is what I really don't like about the thing, but you have to have investors. And they were wanting to get their money out. And so we went public, because that was a time when AgBiotech was a hot thing. And we were the hottest guys in AgBiotech.
So, John, I didn't ask this journey, but I have to ask, like, when you plan to kind of start on your own, like, how difficult or how easy it was for you to raise the money? Like, how did you get in touch with this investor?
I got it without even a plan.
Wow. This is
Well,
fantastic.
I had interactions with some pretty wealthy guys. And so one of them was David Sainsbury. And if you know him, he's now the
I've heard of
chancellor.
him, yeah.
He founded all the Sainsbury grocery stores, and he's now the chancellor at Cambridge. And I had worked with him, and I told him what I was going to do, and he said, "Well, count us in." And I said, "Okay."
Wow.
And then I went over to a local investor who I knew and gave the same story, and he said, "We're in too." And then they contacted me with another investor. And there were 4 investors in the end, all pitching in about $5 million. So
and was it your encyclopedia selling skills coming in handy here?
Yeah, it's always coming in handy, because what that teaches you how to do is how to read people in a room. You have to develop the skill, because people will try anything on you, and they'll test you and stuff like that. And you got to realize what mindset they're in. And I do that pretty well now, but I learned it. It's a learned trait. But yeah, I knew these investors personally.
And when did you do? Because you were in science, right? Like, and this is one of the challenges that all scientists face, because they are between this academic wall, right? Like, they would never get to interact with this investor. So how in the world were you so connected?
Well, we had projects with the companies these guys started. And so they all knew me, like Calgene. The founder of Calgene was Roger Sahlquist, and I knew Roger. And so I had Dennis Doherty out at InterSouth already in, as well as David Sainsbury, and Roger didn't even have to say anything. He just said, "I'm in. If they're in, I'm in." And then we got a 4th guy to say the same thing. And it was just who you were pulling into the deal. And it was all about knowing people, right? And they respected me, and they knew I'd do something good. Well, okay, they weren't going to invest until we had a business plan. And so we spent a month drafting the business plan and showed it to them, and they liked it. And so we launched Paradigm Genetics.
Got you.
But I wasn't afraid. I had 6 months severance because I left during a merger. They didn't fire me, but, it didn't matter. I mean, the company wanted to do good by me, because I'd done good by them for 13 years. And I had a frank discussion with them, and I just said, this is all going to change. And, I'm getting older, and I'm not sure I want to go through that change. And the upper guys understood, and the guys in Greensboro who were our local management, they understood. In fact, all of them retired when this happened. And but they gave me a 6-month severance, so I wasn't worried. And if something had to work out, I could get a job anywhere at that time. And so, no, it was fun. It was really fun. And we raised the money very fast. And then we got setting up the company, and we called it the plant? I'll think of it in a second.
You mean AgTech?
No, the
AgBiome?
Plant Phenotype Lab. That was Paradigm,
Yes.
But then, Paradigm went public, and, the investors, I didn't like them. And I stayed for a year, and I hated running a public traded company. I hated going to Wall Street every 2 or 3 weeks, pitching to some asshole investor who doesn't know what we're doing. And our stock ran from $7 a share up to about $700 a share during the big 6 months it did this. So the company in the year 2000 was valued at $2 billion.
Wow.
And,
Wow.
we didn't, well, that wasn't going to last.
You knew that as an insider, of course.
Yeah. And anyway, I got at odds with a couple of the investors there, and I just left. I just said, it'd be nice if you fire me
Yeah.
because then I could get my severance pay. And they said, okay, well, we'll fire you, and we're firing your wife too. I said, yeah, she'll like that. But anyway, that happened. And
But John, I mean, in retrospect, it looks funny and it looks easy, but forming a company is a Herculean task. It's not that easy. I mean, you make it look very easy,
it
but
was fun and it was intuitive. You know, it's like, okay, I'm like setting up a research group. I'm setting up a business group, but I was also on the board of Ciba-Geigy USA. So I was hearing all the business stuff all the time, and I knew that stuff. I was head of IP for Ciba-Geigy in the USA, the whole USA, including pharma. And so I was well skilled in a lot of this stuff. So for me, it wasn't a big deal. And building a lab was nothing. We'd built labs before. And, so we started putting the company together and refining the business plan. And, within 6 months, we were in pretty good shape. And the investors met again because another guy wanted to come in, and so we got more money. But anyway, we took the company public and the stock rose up to, I think it was $200 a share. Anyway, the company was valued at $2 billion. And it's like, this is ridiculous. And I couldn't sell any of my stock because I was the CEO. And I like, guys, I got to go. You guys can run this company. That's what you tell me. So I got out of that company. Then I started Metabolon.
Sure. You sold your shares or not when you got out?
You can't because you're locked up for a period of time. When they unlocked, I put them in trusts and sold everything. But by then, I don't know if you remember those days. That was in like 2002, and the market was on a run.
Mm-hmm.
Everything was big, but then it crashed.
Yeah,
and we got caught in the crash. So I got out for $7 a share, which still was a lot of money.
Yeah. Yeah, but not compared to 700, right? So wow.
Yeah, disappointing.
Yeah.
A couple of the other found.
But John, this is also like another question because many people think that entrepreneurs, entrepreneurship is all about making money. But it seems like you were not just driven by money, right? Because you were an academic, you were more driven by science or the curious or the fun
I
part,
am
right?
way more driven by the science and the accomplishments we can make for society than I am about the money. If we do good, we'll make money. If it doesn't do good, we're not going to make money. But I don't look at pennies and count pennies, I don't care. And especially after Paradigm Genetics, I had a nest egg of about $3 million, which, for that time, it wasn't that bad.
I mean, gone. Even for this time, it's not so bad, right? So,
yeah, so I left and got back with the guys that started Paradigm Genetics.
mm-hmm.
That's when I had read Fraley's article about the next steps in agriculture was going to be phenotype. And we set out what the phenotype was. And what we really needed was a technology to measure all the small molecules all at the same time. And we'd tell people that, and they'd say, that's impossible. And we'd say, well, we're using mass spectrometry. That's double impossible. There's no way you can do it. And it's like, I can do anything. Screw you guys.
Wow.
And again, two of the investors in Paradigm jumped into Metabolon. And so we had money from the start on that. I took all the guys out of Paradigm Genetics that had been working on that problem. Well, they all just left and they went over to me. And, and that's what we did. We focused on this. It was 10 years of what do they call it? We'd find a problem, we'd solve the problem, we'd find the next problem.
So 10 years of problem solving.
Right. Because originally we could get the data, but the average standard deviation of the data was 50%.
Wow.
And that's why
Nice.
everybody was skeptical. But we started tracking it down to what was causing a 50% standard deviation. And we got it down to 20%. And then because it was different factors in the process. And then we couldn't get any further. And 20%, you could sell it, but that's not great. And we kept working. And my two software developers, Corey DeHaven and Tom Barrett, came to me one day and said, "We know what the problem is." And I said, "What's the problem?" And they said, "It's the software in the electron multiplier." And he said, "Doing crazy shit." And I said, "Okay." And I knew the president of Thermo at the time, and he was on the science board of Paradigm Genetics. His name was Ian Jardin. And I called up Ian and I said, "Ian, we're having a problem with your electron photomultiplier. So we can do this two ways. You could give us the code and we would make improvements to it, and you could have all the improvements you wanted back, for freedom to use." And I said, "Or I'm going to wait till, May, and I'm going to hire a bunch of high school students to come in and decode this shit."
So he gave you the code, of course.
He knew we would do it. He knew me and he knew I'd do it. And so we got the code like 3 days later. And it was true. The code was developed in the 1970s in the early stages of mass spectrometry. And it was doing stuff that you wouldn't do these days, and how you integrate curves and things like that. Well, Corey and Tom fixed it. And, then they put it back on the machine and we got the standard deviation down to 2%.
Wow. So that's tenfolds. Wow.
Yeah. And that's good. That's something
good, yeah.
somebody's going to buy. And so, we just put that in, and that's how we developed Metabolon.
Amazing.
And I still want to start another company, but I can't get any funding for it right now. You got the gray hair syndrome, but
is it just because of the gray hair syndrome, or also because of the market currently?
Well, it's both. I mean, during COVID,
both.
you couldn't launch anything during COVID, and that still hasn't totally passed. But that's those two companies. During that time, two of my ex-postdocs got together with me, and they wanted to start a company for biologics. And I had run the biologics group at Ciba-Geigy. And we had developed some really good microbes, but you have these promotional procedures in these big companies. You have to pass that gate, then that gate, then that gate, and we could never get past the second gate. And so I went over to Basel and talked to the head of business for Ciba-Geigy, and I said, "Look, this is ready to go." And I said, "Are you going to use it or not?" And he said, "Explain to me what would happen." And I said, "Well, you make up the bacteria, you spray it in the fields, and it'll kill the fungi." And he said, "Then it will make our products obsolete." And I said, "Well, you can mix your products in with it and make them super products." And anyway, they killed the project. I said, "The worst thing you can do is to keep scientists working on a project nobody cares about. That is very frustrating for scientists." And I said, "So just tell me." And 2 days later, they told me, and we redirected those guys, and the project was over. And then, that was with Ciba-Geigy. And then when we eventually started AgBiome, because an investor called me. This is a real story. He called me and he said he was an investor in in Metabolon. And he said, "John, have you read the latest Science magazine?" And I said, "No." "Have you?" He said, "Yes." And he said, "There's 2 interesting articles in it that I want you to follow up on, because maybe there's a company there." And I looked at him. It was 2 of my friends, Paul Schultz-Liefert from Cologne, and David, guy at North Carolina University. So we went to them and said, "Do you want to start another company?" come in with me and Scott and Eric. And they said, hell yeah, you know? And I said, we'll just go get those microbes that we had before and we'll just develop them. We knew where we had deposited them. So anything you patent is put into a patent depository and they'll hold it for the life of the patent and not let it out. But after the life of the patent, it's freely distributed. Well, their patents, had been filed in 1989 or something. And by then it was 2009, and so the patents were over. And we started AgBiome around that. We were going to develop that strain. And that's what we're doing now. I mean, one thing that struck me at Ciba-Geigy, and I'll tell you a little bit about this whole thing. One thing that got me was their awareness of when patents were going to expire and things and the steps they would do to protect it, and maybe elongate it. And they did that, but they didn't do it with these microbes. And so we could go in and mix these things with the microbes the way I told them to in the first place and get a great product with a reduced use of pesticides and a long-term protection from the bacteria.
Wow.
And that's what AgBiome was supposed to do.
Amazing.
Yeah, and that's what we took off to do. And they've done it. They did it, but they did some, it was run by two of my ex-postdocs, Scott and Eric. And it's like having your kids run a company, and they ignored the things I was telling them to the point where I just said, do it your way. The company was dead 3 years later.
Why was it dead?
They ran it into the ground. They were arrogant. They were very arrogant, and they were ignorant because they didn't understand the principles of business. But they thought if they asked for something, they'd get it because every time I asked for something, I'd get it. But they weren't me.
Yeah, exactly. You cannot copy. Wow. John, coming back to Metabolon, what are the challenges that you faced while building Metabolon, and what are the current challenges that you see as now like an external person?
Well, there were many challenges building that thing. And, we had to whack-a-mole the whole time because you'd fix something and then something else would come up. We're talking Metabolon, right?
Absolutely.
And so we finally got, standard deviation down to 2%. And we knew how we did it. And we had investors that didn't understand a thing about what the company did, which I would caution all inventors and entrepreneurs to be careful with the investors you get, because if you get investors that don't know what the hell they're doing, they're probably going to be bad investors. And you got them until you don't. So anyway, what were you asking about Metabolon?
No, I was asking what were the challenges that you faced? Like you said, one was the standard deviation thing. Apart from that, you said the investors' priorities perhaps changed. And
we had to identify the compounds. And to exactly identify a compound, you have to have a standard. And so we had to go out and source 2,400 standards from companies and from universities who had them in order to get a standard sample to run through the system and say that's what that compound is. And then we developed software to go through the waveforms. I don't know if you've ever seen the data of a mass spec. It's about as thick as a ream of paper. And so these algorithms would go through that and find that peak, and identify it. And then it would quantitate it, using a certain quantity. And we developed quantitation methods called choosing or voting. And so the computer would use 4 different ways to figure out a curve. Then the computer would look at the 4 different ways and pick the way it wanted to do it, and then it would do all samples the same way. So that was a development, and that was a huge development in this theory and everything. Having no-touch samples, because much of the original variability was coming from when people touched the sample. And if you got 20 people passing on a sample, maybe somebody's come in with a hangover, And there goes your standard deviation. But even if they don't have a hangover, they're going to screw up your standard deviation. So you have to get the people out of the project.
So you mean like one can build a system where like you don't have to even touch anything, and then that is possible. One
Yeah,
can
we did
build.
it at Metabolon. And now we want to take those principles and build a new instrument where all you got to do is inject a sample and you get the results. And nobody can do this, because the gatekeepers of metabolomics are mass spectrometrists,
Mhm.
because you got to use the mass spectrometer. Well, they're not easy to use machines. People go to college for 4 years to try to figure out how to run one. And, so the idea was to take the mass spectrometer off the table
Exactly.
and democratize the technology. And we talked earlier about DNA sequencing and automated DNA sequencing. And the guy, so when I first got to see the guy, they were doing stuff silly, and I came out of the best. You couldn't do phosphotriester synthesis because if you had any relative humidity, it would cleave the phosphotriester. And we knew that in Switzerland because we bought one, put it in a dried nitrogen cage, and it worked fine. But that was a problem. But then phosphoramidite chemistry came out, and that was like, okay, and that's stable. And so in DNA synthesis, we were using phosphoramidite. Well, I told the DNA guy at the company, he reported to me, his name's Steve Lombardi. And I said, he came to me one day and he said, I know what the problem is. And I said, what's the problem? And he said, and I don't know the chemistry well enough, but he said, this and this. And it's just not working. And I said, interesting. And he says, they designed it wrong. And I said, interesting. Do you know how to fix it? And he said, yeah. And I said, well, I'll buy you a ticket out to, Applied Biosystems and get somebody and fix it. And he did. He fixed the problem. Applied Biosystems already knew they had the problem. And Steve came in and in like less than a day had totally solved it. And Steve never came back. They offered him like 3 times the salary we could offer him. And they made him the head of the synthesis group and then the head of DNA synthesizer group.
Fantastic, yeah.
And so I'm looking at metabolomics and, Steve's still a good friend of mine. I mean, I wasn't pissed off. When he told me, I just started laughing. That's good for you. You're doing something you love now and you're getting paid what you should get paid. And anyway, so me and Steve have always kept in touch. We've been friends for 25 years. No, 40 years. And so we had this problem with with metabolomics. And we had this idea of building an instrument. Well, we had an idea of how they could build an instrument to do metabolomics right. And nobody would take it. Not ABI, not,
Bruker,
all the companies.
yeah.
Bruker, nobody. Thermo. And I was very, very confused. And I went back to Steve and I said, why aren't these guys jumping on this shit? They can make billions of dollars. And he just said, it's not their business plan. And I thought about that for a while and I thought, really? But discovery in this stuff wasn't part of their business plan. And so he said, what you're going to have to do is build it yourself. And I said, build a automated metabolizer. And he said, yes, one touch. And he said, yes. And then people will buy that. And he said, that's what we did at Applied Biosystems, with the DNA sequencer and then the DNA synthesizer. And as long as they followed our menus,
Instructions,
it would do what it did. And, and they would put standards in the stuff. So they went and troubleshoot and they didn't see that standard in there. Then they knew they didn't use the right equipment, the right liquids. And so we took that approach in building this thing. And so I had the mass spectrometrist that had developed Metabolon because after I left, most of the really good guys left. I mean, the sorry situation was that Roe was not the right guy. He was vibrant and he had interpersonal skills and stuff, but he didn't know business very well. And the first thing he did after I left was to cut people's salaries. And, I had set their salaries at like the 75th or the 80th percentile because we had the best people in the world.
Mm-hmm.
Well, he had them cut it to the 50th percentile. And some people just left in droves. This is probably before you went there.
Mm-hmm. Mm-hmm.
But anyway, we picked a lot of those guys up. So we got the mass spec guys that can build it. We know how to source it. Even Applied Biosystems and Thermo source all their shit from China. So you just source the shit from China. And I've got a very good Chinese friend who's the best chemist I've ever seen.
Wow.
Lenin Vo, and he's with us. And he said, yeah, I can go source this stuff.
So are you working on this actively, John?
Well, I'm going to reborn it again. We tried to start it before COVID, and it was just before COVID. It was like in April when COVID started. And as that developed, nobody was going to fund anything, and even for 2 years, nobody was going to fund anything. That's just the way it is,
Yeah.
And now they're still reluctant about stuff. And when I tell people, what I need is enough money to build a prototype so people can see we can do it. And we need $2 million to do that because we have to actually buy the instruments because we're going to tear them apart. So we can't just lease them. So we're looking for that seed money, and we could build it. I think we could build it in 6 months. And we have the software developers that already had the software. Because they stayed in metabolomics, and we got the analytical chemists that'll come along. We got everything. And so that's what I really want to do because I spent so much time in my life doing this. And to see it being held up by a bunch of mass spectrometrists really pisses me off.
So a message to the investors listening out there, you have a business case here to invest. Wow.
Yeah. So leadership, you want to move on? So leadership and lessons learned.
Yes.
Yeah. So you say I've been called a failed retiree. You must talk to a lot of people.
Yeah.
Because that's what I
But now,
call myself.
now midway of the conversation, John, I can always see that you're not a good person to retire because you're so active mentally, right? So, I mean, you got to do something, of course.
I had to retire because of stress, and these venture capitalists on 3 companies were beating me up all the time. And after a contentious board meeting at Metabolon, I went incoherent, in my office or at home. And a couple of people had to come take me into the emergency room. And I had a blood pressure of 240 over 220. And the docs said, because I know a lot of docs over there, and they came in and said, we're going to tell you, you got to stay here till we can get your blood pressure down below 150. And so I had to stay a week or so. And they said, but we're going to tell you, you had 2 choices. You can continue to work and you're going to be dead by the end of the year, because of all this stress, or you can retire and it'll all be gone, And so I got a younger wife who I love with all my heart. She's
and I should have asked, how did you meet your wife?
How did I meet my current wife?
Yes.
At the what's it called? It was at the Raleigh Art Museum Halloween ball, and I was a conquistador, and she was a sexy witch. You're an absolutely beautiful, sexy witch. But we have a common friend, a Greek guy named Thanos, and Thanos introduced us.
And when was this? And when was this, John? When was this?
2002? Was it 2000? It was 2003.
Got it.
But it was love at first sight for me, and she said it was love at first sight for her. And it's like that movie, one enchanted evening, you'll see a stranger across the crowded room, and then you'll know.
You'll know. Absolutely. And I can feel the vibes, John. But coming back again, so John, on one side, I see you are so passionate about what you have done, and normally I hear that, stress comes only when you do things that is against your will. And you have
Right.
always pursued things that is, that you wanted to. What was the reason? Was it just the investors, or what was the real reason of all this stress that you had?
Yeah, as the investors, and after I quit, I was on the board of these companies, so Metabolon, I was still on the board of AgBiome
Mm-hmm.
and another board. The other board didn't bother me, but I would get on the board meetings. The virus disease?
Mm-hmm.
It was in the middle of that pandemic. The
COVID. You mean COVID? Yeah, okay.
Yeah.
COVID. Yeah, okay. Yeah.
And it was in the middle of COVID. And what was the question you asked?
No, I said, what was that was stressing you so much?
Oh, well,
No?
like I said, it was those meetings. And so I went home, but I stayed in those meetings. They were, pretty soon they were done, by video, because of COVID.
Mm-hmm.
And my wife said every time I got off a board call, I'd be
Your
a different
blood pressure
person.
was high.
Yeah, I was a different person. And then she went and bought, you gotta love her. She went and bought a blood pressure machine. It's right over there. I'm not making fun of you, baby. Just saved my fucking life.
No, of course.
And she would put it on me before I got on the board meeting.
And she measured, like she did the experiment, basically, right? Control versus.
Yeah, and I go from my normal blood pressure's 125 over 70 or 75, and it would quickly rise to 140, 160, 180, and then she'd show me and make me hang up. Okay.
So, John, I mean, on one hand, I see that, investors gives all this stress. On other hand, you never bootstrapped. You always relied on investors' money, right? So how would you kind of justify this?
Well, I don't have enough money to launch a company, and
Mm-hmm.
I mean, it would take us $2 million, and I don't have an extra $2 million until Metabolon until Metabolon liquidates, and then I'll be fine.
No, so coming back, so no, I understand that. Of course, sometimes you don't have enough money, and that is why people go to investors. That's all. But the thing is, how would you advise others? Like sometimes I have seen many successful entrepreneurs who never went to investors. For example, this evening I was talking to somebody in Silicon Valley, and she said she has a company which is like 50 people company, and she never wants to grow much bigger. She said, I am very happy and satisfied person, and I don't want to have others' money and have all the stress. So what's your take on this, looking back?
I can understand her, but here's the deal and why I went into industry. I didn't say that because I didn't think to say that. I have big ideas, really big ideas.
Mm-hmm.
And that takes a ton of money to do.
Yes, true.
And where do you get a ton of money? You're not going to get it out of the university,
True.
because to do one of these things, you're going to need $2 or $3 million a year, and you're not going to get that from NIH or from the university, so that's why I went the private route,
No, makes sense. And what would be your advice to, the young scientist who wants to be an entrepreneur? How would they go about building a company?
Call me.
Of course.
Seriously,
Of course.
It's not a one-word answer. It's complicated. It could be a whole different talk, but I'll help anybody, and so if they want help, I can teach them. I mean, it's not as hard as they think.
Wow. No, this is so nice. So coming back to this leadership lesson, so what were your leadership lessons that you learned all this way?
Well, growing up, I never had anybody report to me like as a postdoc,
Mm-hmm.
but by the time I got a permanent job, I was a lab leader, so that was the first time I managed people. And I had to learn how to do it. And my dad was still alive at that time. And, and I'd call him about things. And I would think about how Guy Spuler had done it, and how he'd never told people what he wanted. And so I kind of adopted that style, and it's very, very effective, because you get them to formulate the question and formulate the problem. And you sit there when they formulate it. But when they've gotten to where they need to be, you stop and say, yeah, go solve it. Number one, that empowers the employee. they feel great about it. I didn't tell them to put 10 milliliters into a petri dish. And, they did it themselves. And since they did it themselves, they're even more proud, you know?
Yes.
And I think that's a very good leadership style. There's somebody in the triangle that does that I ran into the other day. But anyway, that's what I learned from Guy Spuler. And I tried it out at Ciba-Geigy and it worked really, really well. I was always the top manager of the year for research, because I was a nice guy. But, and if they screwed up the first time, I'd say, okay, you got to pass. The next time we're going to do this more seriously. And then they'd do it again. I'd let them formulate the question.
Mm-hmm.
And if they formulated the question wrong, I would say, no, that's not right. This is what you need to be looking. And they said, oh, I need to do this. And I said, yeah, go do that. But I wouldn't tell them how to do it. They could ask their friends or they'd do it themselves. And what you're doing there is you're enabling the employee, and employees love to be enabled.
Absolutely.
They don't want somebody telling them what to do all the time. And you're growing their own management skills and their own thinking skills. And, I'd give them 3 chances, and if they did first chance, okay, that's first one. Then on the second one, I would sit with them while they formulated what the problem was. Because you don't want to do anything until you got a very well formulated problem, so that you know exactly what the problem is. And I taught them that, and then they'd go away. And most of them I never saw again. They just did it, and then they just adopted that. But occasionally a person would come back again, and I'd say, huh. I said,
no, this is
maybe you should go somewhere else because you need to be somebody's technician, not a scientist here. But that was actually my leadership code, and numbers of people picked it up after me.
No, this makes sense. This makes sense.
And it works the employees love it. If you're making all the employees' decisions, they feel unempowered. They feel uninvolved and they don't like it. And I've seen people that just stand over an employee, lab worker, and just beat them down. Why didn't you know this? Why didn't you know that? And it's like, that's not going to make somebody learn. That's going to make somebody scared. So I just don't know that. And I learned that. That's my style. And it's becoming the style of a number of companies because people that worked under me are now going to other companies, and they're employing it in the same way. But that was the Swiss style, And in that last talk I had with Guy Spuler, I asked him the question again, and he said, John, you know the answer.
You had to discover the answer yourself, right? He never answered you. He never told you, yes.
And I said, yeah, I know what it is. And I told him, I said, you're making them reach the decisions themselves where they will learn instead of telling them the answer because you're being, and he said, he nodded his head. That's all he did.
No, I'll keep this in mind as well. This is fantastic.
I love that, man. I love that, man.
Coming back again, what do you think has been the biggest lesson about scaling biotech companies?
Yeah, well, don't do it faster than you need it. Have the money to do it. Don't get halfway in and need to raise money because investors will take advantage of you. If you get halfway through and you say, I need money to finish this thing, and the investor goes and sees the half-finished lab, you're not worth as much as you were because you're needy. So
that would be
I would keep that in mind.
When you reflect back, what do you think are the mistakes that you made in your entrepreneurial journey, basically? The mistakes.
Most of the time, I didn't communicate clearly enough to the person, and they didn't get it. And I didn't get that they didn't get it, and that's a problem.
Miscommunication, basically.
Yeah, and generally that's the biggest problem I ever had. Is people, you can't assume they're going to understand what you told them the first time. And you can always get them to repeat it back to you. But you don't want to do it for them. And I think that's very important because you're building the employee and you're teaching them.
Yes.
While they're helping you out. you know?
Great. So coming to, the next section, basically I'll be asking you, how do you see the future of metabolomics and how do you see the future of AgBiome? Both.
AgBiome, I don't see a future in AgBiome. No, they went bankrupt last year.
That's not a very
They
good
just closed down and dissolved the company. No warning to
anyone.
anyone, to employees or anything. They got into private equity, which I told them not to do because they didn't understand what private equity was. But of course, they were both ignorant and arrogant. And that's
A deadly
not a
combination.
good pairing. Yes, deadly combination. You can be ignorant. You can be arrogant, but don't be both.
And how do you see the future of metabolomics and maybe also metabolone? How do you see that?
Well, I've offered this idea of making the instruments to Metabolon, and they turned it down. So I may go back to them again, but I think they have a certain bit. They're sitting on $1 billion database. Now, they've done tens of thousands of studies, and they know what the study was because they had to interpret it, and they know what the markers were. So they could mine that database like hell. Or they could sell it, they could lease it to somebody who could mine it like hell. And I've told them that over and over, but they won't do it. And it's just like, what do you think every other company does? Every company does that. So I don't know future Metabolon. I hope they do something. But I got a good share of that. What's the next one?
And the future of metabolomics as a whole, because, there's with advent of AI and stuff, there's so much things you can do, right? So,
well, you got to be careful with AI. AI is a statistical method, so it's got statistical limits that are set, so it could be wrong by, you could allow 5%, you could allow 10%, but that means 1 out of 10 of your findings is noise. So people had better be aware of that. I mean, it can do these tricks it does, but if you pull it into real science, because I know a company that's trying to do AI on mass spec. I'm like, okay, give it a whack, see if it works. And I think,
wow. No, you're right. You're right. There's a lot of hallucinations in AI, also like in normal day-to-day applications.
Well, if you're a statistical person, if you set the standard deviation at 10%, 10% of your findings are going to be wrong. That's the way it works. And I don't know what AI sets their standard deviation at. I mean, I think AI is going to be most important in precision manufacturing, where things are very, very precise, but maybe not so much in other things. But what do I know?
Yeah.
And my wife's in AI.
So, yeah, go ahead, John.
No, what do you want to move to?
No, I just wanted to say that I wanted to do the rapid fire before I would ask the advice for the students and others, so I would rather prefer the rapid fire first. All right.
Let's do it. Let's go.
Let's do it. So there's question that I will be asking you, but you don't have to think about it. You just have to answer at the first instinct. So favorite scientist of all time?
Modern science?
Any scientist.
Maybe Edison.
Thomas Edison. Perfect. A biotech innovation you wish you would invented?
DNA sequencing.
What is your go-to productivity hack when you are deep in research?
I don't understand that question.
So it's like productivity hack, what do you do? Like many people they have a coffee or they have yoga or meditation or they just go for a walk. What makes you more productive?
I may walk around the building, but I usually just sit there and stare at it, And that works.
Okay. So if you weren't in biotech, what career would you have pursued? Yes. No, in the beginning itself, suppose you did not study biotech, what would you have done? What was your second?
Well, I was going to be a professor.
I mean, yeah, this is true. But my question was if you didn't pursue biology altogether, you would be, I was thinking that you would be a musician, but yeah.
No, I think my fallback was to be a lawyer.
Okay. Wow. Okay. Lawyer. Wow. I never thought of that. Cool.
Well, by the time I was majoring in music at college, it had turned into that disco crap.
Yeah.
And it's like,
you had mentioned that. Yeah.
I'm not going to do that.
Yeah. You mentioned
it's bullshit.
That.
There was disco, bar right across the street from my apartment.
And John, you had mentioned that two books like The Ascent of Man and The Molecular Biology of the Gene was like very influential. But after that, what were your most influential like book that that inspired you?
That's an interesting question. Well, molecular biology of the gene dumbfounded me of what they knew about DNA and stuff.
Mm-hmm.
Because I was still not in grad school. And, I read the book and then I was committed to molecular biology. The Ascent of Man, I think that's influenced me from, so that was there was a TV, you ought to get on PBS, files and find The Ascent of Man. I think it's still there.
Mm-hmm.
And it was a British series in public broadcasting. And there was a guy, Jacob Bernowski.
Mm-hmm.
I thought that was right, but I didn't know it was right. So he wrote the book and he narrates the film.
Mm-hmm.
And just the way he would dig into problems, and, he'd start off by showing a problem and explain why it couldn't be solved, and then he'd spend the rest of the hour solving it. And I thought, hey, that's great. And he dealt on really interesting subjects. So I was into archaeology and paleontology and stuff like that. Yeah, I've been into all that stuff. And listening to he got into writing that book because he was a professor at Harvard and a mathematician. And one day a student came in with a human skull and he said, how can I measure the volume of this skull? And Jacob Bernowski said he stared at that thing for 2 days, then it hit him, and then it convinced him. And I can totally understand that. He solved a problem that he couldn't solve. And then he thought, okay. And he got interested in archaeology. And so that's what he did.
Mm-hmm.
He had a full professorship at Harvard and he left. Yeah. You get the series. It's called The Ascent of Man.
Okay, moving next. So one piece of advice that you would give to your 20-year young self.
Have a lot of fun.
You had it, John.
Well, that's my advice. I did it and I had it. But yeah, don't get too serious about things and serious about yourself, because anything can happen.
Mm-hmm.
Anything. When I look at the things by chance that just happened, number one, you got to realize that just happened because a lot of people don't realize they don't have the epiphany, and you need to have the epiphany.
Mm-hmm.
So I think the epiphany is one of the most important things in logic. You're shown stuff other people have seen and nobody could solve something, and all of a sudden I see it. And so, and don't discard the epiphany. Some of my greatest discoveries were because of my epiphany.
Fantastic. No, that makes sense. All right. So coffee or tea while working in the lab?
I had Diet Coke.
Fantastic.
And I don't really like coffee or tea.
And one word that sums up your journey in science so far.
Sums up I would say amazing, talking about how I view it. It's amazing. If you told my 14-year-old self that this is what I was going to do, I would think you're out of your mind.
Yeah, now coming back
and it's been fun.
Yeah.
Yeah, it's fun.
Amazing. Fun and
amazing. It's fun to break the standards.
Absolutely.
It's fun to say that's not really how it happens. And I've been privileged to be able to do that quite a few times,
multiple times.
And so my research from the 1970s is still being cited. My research from the 1980s is still being cited. So I don't know if you know how they judge academic scientists.
I understand. I am a scientist. Yeah, all this citation or whatever H index. There are so many ways.
The H index. So I've got 38,000 citations on my papers, and I've got an H index of 80. That's pretty high.
That's pretty high. And especially that you didn't you were also an entrepreneur. So you were not like a professor, right? So that's pretty high for a...
And I'm very proud of that.
You should be. I mean, John, this is like...
And I've got papers I published in the late 70s and early 80s that are still being cited. They're seminal. I actually Googled seminal the other day, expecting it would be some sort of rosary. And the answer wasn't satisfying. Seminal to me means that it's there, it's true, and it's there forever.
Mm-hmm.
And a lot of my research is now deemed seminal.
Wow.
And for scientists to have published a paper that's considered a seminal piece of literature,
This is a great feeling.
yeah, I think that's amazing. And so I check my citations on like a daily or weekly basis just
Wow.
to make me feel, just to give me a warm feeling, you
No,
know?
no, I can totally understand this. It's like,
Still,
wow.
I'm still accumulating them.
And rightly so, well deserved. So, John, like if you were to advise, because a lot of the viewers of my podcast are actually PhD students or, postdocs who are kind of looking forward to establishing their careers. So what would be your advice to them?
Well, I would tell them to work hard at whatever they choose to do.
Mm-hmm.
Don't be some part-time worker.
Mm-hmm.
Learn everything there is to learn and to do something that makes them happy.
Mm-hmm.
That's probably the most important thing.
Mm-hmm.
Don't work on something you don't like. Then 40 years later, you did something that you didn't like.
Exactly.
And I would tell them to follow their nose and follow what they enjoy, make them enjoy it, make them happy.
But sometimes, so this is, of course, this is a very common advice, but sometimes, this doesn't, like sometimes you're forced to do what you don't like because you got to pay the bills. So not everybody's as kind of, I would say.
That's okay. That's okay. But don't do that the rest of your life.
Mm-hmm.
Find a way out.
Mm-hmm.
I had friends from high school. I graduated in 1972 from a place called Carter High School, which is one of the schools in Friday Night Lights. I remember those football games.
Mm-hmm.
I couldn't play football back then because I was 6'2" and I weighed 135 pounds. I went out on the field. And one of the coaches came up and he said, "You see that guy?" He says, "He's 6'2" and weighs 350 pounds. He will kill you." So they wouldn't let me play, but I found fun at other ways. But yeah, I mean, once I got into it, I realized it was a great move.
Mm-hmm.
And I realized I did right. And so there was no need to question or change. I was doing what made me happy.
So you would always advise this, that, no matter what, always do things that makes you happy, because ultimately that counts.
Yeah. And if you have a dream job,
mm-hmm.
Well, there may be steps to get there that will make you don't have to pick up the garbage.
Mm-hmm.
You can do other things to get there. You can go work in a university,
Mm-hmm.
They hire all kinds of people, but don't give up your dream and work hard. Work very, very hard. Work until your shoes fall off.
Wow. No, fantastic.
Absolutely.
No, absolutely.
I mean, in my early days, I'd work until midnight, and then I'd come back at 7 o'clock in the morning. That was when I was in my 20s but I did work hard.
Absolutely. And still I can see that, I can see the energy, John, right? Like the first call that I made to you was already, I could feel your vibes. When you said like, "Oh, Manish, my friend, my new friend," I was like, wow, he's a charmer,
Well, you are my new friend.
Yeah. So, John, when are you
nice emails.
When are you visiting Germany, John, by the way?
Where in Germany are you?
I am based in Mannheim, near Frankfurt.
Yeah, I know where Mannheim is.
It's not very far from Basel or Zurich, actually. So you can also visit Guy Spiller if he's around there.
He died.
Sorry to hear that.
Yes. He died 2 years after I had lunch at his house. And I'll tell you, I cried.
Of course. I mean, I can totally understand. I feel bad now, because, of course, this podcast was a lot about him as well, right? Because all the lessons.
He was a great man.
Absolutely.
He was a great man.
Mm-hmm.
Well, I got a lot of European friends.
You should just come and visit. And then that's friendship. And then we go to a, cruise from Mainz to Koblenz. And I see a lot of Americans enjoy that, because that's also my favorite part to do in the summers, John. And you will love it.
I did that. I was in Europe for 2 years living there. And then I was visiting there at least 2 months a year for 13 years. You're working for a Swiss company. So I know Europe pretty well. But I like it, And And I've got friends over there.
You see?
It's just
So all the more reasons to. So John, thank you. I have to respect your time as well. You have been very kind. I really enjoyed the conversation with you, and I hope the audience will find a lot of value and inspiration from this podcast. Yeah. Thank you.
Well, thank you. Very nice meeting you, my friend.
Same here, John.
And hope to see you in person.
Hope to see you in person for sure. All right.
Okay.
Thank you.
Bye-bye.
Bye.